Okay. So can everyone see my screen and slides?
Yes.
Okay, cool. Thank you. So, okay. Thank you so much to the organizers for giving me the chance to speak today, as well as the PEB folks at Yale for allowing me to give a talk alongside Professor Sweeney. So my name is Jack Treado. I'm a PhD student in mechanical engineering at Yale and part of the physical and engineering biology program, which is the Yale hub or the Yale part of the IPOLS network. I'm really excited to be talking about some new research that I've been doing on characterizing and understanding development of this complex tissue called the Spongy Mesophyll that's found in plants. And this is work that I've been doing recently with my advisor, Corey O'Hearn, as well as our collaborators in botany and biology, Adam Roddy, who's at Florida International University, Craig Broderson, who's at Yale in forestry, and as well as our physics collaborator at CCNY, Mark Chaddock. And this, I'd like to also acknowledge my funding from the NSF. So before I dive into this very complex structure that I'm showing here on my screen, I want to take a step back and first talk about the organism that made that structure, which is a plant that makes flowers, or a flowering plant, which are generally known as angiosperms. And so the angiosperms are by far the most dominant terrestrial type of plant. Most plants that live on Earth make flowers. And this plant here is the flowering dogwood, or the Cornus florida, and this is actually the plant that made the structure that I was just showing you on my previous slide. That structure is at a very tiny microscopic scale, but if we just think about what do flowers do on a somewhat larger scale, their main function, of course, is to house the reproductive organs of the angiosperms, and not only to house the organs, but also to promote them and advertise them to possible passing insect pollinators. So you want a flower that's bright, but also is a nice stable surface that's mechanically rigid, that the insects can see clearly and can land on to gain access to the pollen produced by the plant. And then another important function of the flower is that it's short-lived. The plant isn't going to be spending a lot of expensive carbon and water resources on maintaining this structure. It's only going to be around during the reproductive season and will wilt and fall off the plant once the reproductive season is over. So these are the functions that sort of define what the flower has to do. And so if we think about the forms that will allow plants to do these things, that doesn't really constrain the space of possibility all that much. All you really need in terms of form to satisfy these constraints is that you need a platform that's mechanically stable or rigid, and it needs to be, in some sense, inexpensive. So the plant shouldn't be using a lot of its precious carbon and water resources to maintain this structure. So these are the sort of general principles that determine everything about flowers. So now let's take a little deep dive into what the microstructure of these of the tissues that make up flowers, what it actually looks like. So the tissue that I'll be showing, and what I was just showing on my previous slide, is something called the Spongy Mesophyll, which is basically just the tissue that lies between the two epidermises of a flower petal. So it's just the interstitial tissue that makes up the flower. And so the Spongy Mesophyll tends to look something like this. This is a sample that was taken from the flowering plant viburnum. What we're looking at here is something called a micro CT scan, or from micro X-ray computed tomography, which is basically just a CAT scan taken on a living flower petal with a very high intensity X-ray beam. This was acquired by our collaborator, Adam Roddy, who's now at Florida International. And this technology allows us to get resolution in 3 dimensions of this tissue down to around 3 microns. And we can resolve basically anything that is cell, because in the interior of flower petals, the cells are mostly water and a little bit of collagen, and the interstitial space is air. So there's good contrast, and we can binarize the scans to really see the difference between the interstitial space and the plant cells themselves. But this is not the only way that a flower can solve the constraints that I was just talking about. Oh, also, we can sort these things by packing fraction, which is just amount of cell material divided by the size of the sample volume. So if we go a little, if we look at a slightly different species that has a little bit denser packing, it looks totally different. So now it's not this isotropic porous network. It's still porous, but there's sort of alignment of the cellular material in a specific direction. If we go a little bit denser, we have this Philadelphus, which is also aligned and even denser. It's almost like you had a bunch of cells that were really long pencils, and they were all pointing sort of along a specific axis. And then as this sample rotates, we can see, you know, this is the back of the pencils, and now we're looking at the side. So it seems like, oh, we go denser, the different species tend to become more aligned, but that's not necessarily the case. So this is a slightly smaller sample, but it's from a species called Toxicoscordion, and it's maybe you can see a little bit of alignment, but it's really reminiscent of an isotropic network, even though it's at a denser packing fraction. But just to confuse you more, we have this very dense sample of Elysium that, again, looks like we have alignment along a certain direction. So we have a broad diversity of possibilities. There are many ways that plants can solve the constraints of having a mechanically stable and somehow resource inexpensive flower petal. This diversity is going to be our task in trying to understand how these different structures are made and also just how to characterize them. But just to make a little point about comparing these things to actual materials, if we were going to see these microstructures not in a flower, but somehow in some inorganic material, these samples down here, they would typically be solids made of attractive particles, going up to about a packing fraction of around half the box volume being filled with stuff. That's typically the realm of gels or colloidal gels, even down to these low packing fractions. This is like something like a hydrogel. In this realm where we have no samples, this is typically where hard particle granular materials live. And then at these very dense samples, this is where we would typically have things like foams or emulsions or dense epithelial tissues. So we have basically the broad gambit of possible soft solids represented in the different scans that we can obtain by just looking at flower petals. Okay, so in my talk today, I want to address two important questions about these tissues. One, how do we think about them? How do we characterize them? They're structurally complex and diverse, so how can we start to tackle understanding those structures? And then thinking about not just what those structures look like, but how did the plant go about making this? Can we construct our own model that would recreate the developmental process? And if we can make such a model, how simple do we need it to be in order to reflect the important features of such a complex tissue? But first, I'll talk about characterizing and understanding the structures themselves. And then in the next part of the talk, I'll go on to development and modeling these things. Okay, so in order to understand the experimental data, there's lots of different ways you can understand structure, but you can't do much better than looking at correlations. So what we're going to do is we're going to compute the Fourier transform of the density autocorrelation function. And if you take a spherical average, you get something that looks like this, which is a structure factor. And what the structure factor tells you is that at a given wave vector k, which is an inverse length scale, how correlated are how correlated is the material at different length scales. So there are a few features that you can glean from a sample like this viburnum just by looking at this structure factor. So one thing that you might notice is that there's a well-defined power law decay at high wave vector or high k value. And this comes from something called Porous law, which just says that if you have very sharp interfaces, then the scattering function off of those interfaces is going to go like something of 1 over k to the 4th. So what we're seeing here is very low length scale behavior of just the fact that we have a binarized sample. And so this is just reflecting the sharpness of the interface between the cells and the airspace. But we can also look at something like the first moment of these wave vectors if we think about the structure factor as a distribution. And so this first moment thing that I'm going to call k bar, if we look at where this is situated on the plot, it's somewhere right in the middle. It's sort of separating this flatter region between the flat region and the region where you start to have power law decay reflective of the interface. So this higher k stuff is going to be physics and structural information happening at very small length scales. So then the business end of the structure is going to be more at these small Ks, which is going to reflect structural features between cells and across the entire sample. So that's good. So we can start to compare different species. So I have 16 scans that were acquired by our collaborators. And if I plot them all together, I can scale them on the same axis just by dividing by the special wave vector. But what we can see when we compare them is that there's not much to really differentiate them just by looking at the scale. I mean, they all have this 1/k⁴, which is just because they have very sharp interfaces. But if you look at this lower K behavior, it's kind of a mess. I mean, maybe there's a few that have some peaks, but there's not a lot of really well-defined peaks in the structure factor, which tells you that there's not a lot of really well-defined length scales in these different samples. Maybe this one does or this one does, but on average, they typically have a smearing of important structural features across many length scales. But one thing we can do is we can just compare what this first moment of the structure factor is telling us by turning it into a length scale. So a wave vector is 1 over a length, so 2 pi over that should be an actual length. And this is going to be something like a correlation. And I'm just going to, correlation length. So I'm just going to sort this by the packing fraction of the sample from which I attained the correlation length. And there's not a lot, I mean, we don't see like a trend or anything, but there are some interesting features to highlight. So for example, we have a big outlier that has a huge correlation length. And that's the sample that I was showing before, which is this lily. And this large correlation length is reflective of the fact that you have these tubes of cells that extend across the entire sample. So that's one interesting structural feature. If we look at some of the things at lower packing fraction and lower correlation length, we have these two samples, which are sort of like isotropic porous networks. A little larger on length scales, and in the packing fraction, we get back this Philadelphus sample, which looks like oriented, long, aligned cells that extend in a certain direction, even though its correlation length is still less than this lily. And then if we go into the denser samples, we can see, again, this is it's got L bar, you know, it's sort of maybe between this value and this value, but it's definitely a little higher up there. And these things are certainly aligned, even though the packing is the sample is very dense. But even the densest sample, we can still have decreases in this correlation length, like we have in this Calycanthus sample, which is basically, it's a very dense smattering of cells that don't seem to have any directionality to them. So this seems to imply that if you start to get larger correlation lengths, that might imply that there's more alignment or more order in your sample. But remember, the structure factor that I was using was an isotropic measure of correlations. But alignment is an anisotropic phenomenon. So we can't really rely too much on getting anything more from the isotropic scattering, or the isotropic correlations. So we have to look now at anisotropic correlations. So again, this told us some interesting things about the samples, but it's an isotropic measure of correlation. So we really need to look at something anisotropic. So of course, this correlation function is three-dimensional. I just took a spherical average to show you this plot. But while visualizing a three-dimensional function is kind of difficult, we still need to do it in order to understand what are the different structural features in different directions from our samples. And so we can do that by looking at something called isosurfaces, which is, you can think of it like the three-dimensional analog of a contour line. So what I can do is I can plot the set of all of the wave vectors in my space that satisfy the function equaling to some iso value S0. And so here on the right, this is going to be the three-dimensional correlation function in all of its glory, just for increasing values of S0 for this viburnous sample. And so scanning through, you can see that the shape of this function, it's not changing too much, but this is going to tell us something about what this how this sample is correlated at different length scales in different directions. So first, I want to look at the samples that by eye to me look isotropic. So this is the viburnum, this cornice that I showed at the beginning of my talk, and also there's this very beautiful sample from a magnolia. And then if we just look at these three isosurfaces, for these three samples, as we scan through them, we see that they're basically doing more or less the same thing. Okay, maybe this isosurface is a little bit non-spherical, but they're pretty much all staying the same. The surfaces are all staying the same geometry as you scan through. And that reflects the fact that these are all isotropic samples. And basically we could have just used the isotropic average to understand the structures of these things because there's not a lot of alignment or anisotropy in the structures. But we also have lots of samples that we've noted do have anisotropy. And just like all happy families are the same, but unhappy families are different in their own way. Same thing with anisotropic structures. So if we go through these one by one, the lily, for example, if we scan through this isosurface, we'll see that it's not exactly, sorry, let me do that again. It's not exactly a sphere. It sort of has different undulations as you scan through it, and it ends up being very flat. Same thing with this Philadelphus. And oh, look at that. It actually gained a hole in the isosurface. It didn't just stay a sphere. It actually, the genus of the surface changed as a function of scanning through. And this Elysium is basically a flat pancake all the way through. Now, this flattening of the isosurface reflects the fact that there's elongation in the z direction, which is, it's a little confusing, sorry, but it's sideways in the image. It's sort of pointing along the elongation direction in the images, but in the scans, it's the vertical direction. And so this is flatter in the z, and these are flatter in the z direction because you have elongation in that direction, which in wave vector space means that your correlations are going to be at smaller k's in that direction. So that's what we're seeing by flattening out these isosurfaces in the z direction. So that also agrees, I think, with intuition. And just to kind of group everything onto one mega space of possibility, we can look at the packing fraction of samples, their wave vector, and then this alpha max, which I'll just define as, it's essentially the aspect ratio of the isosurfaces, the maximum aspect ratio of those isosurfaces as I scan through the correlation functions. And what we can see is, again, there's not like a nice line or something that hints at some deep trend, but we do see pockets of possibility. We see a lot of samples that have small correlation lengths. They're typically porous, and their isosurfaces are typically spherical, which suggests that these are like the isotropic porous network. So that seems to be a dominant phenotype that we see in a bunch of different diverse sampling of species. Whereas we have some notable deviations, like we have this sample of the lily, which has a really high correlation length, even though it's sort of porous. So understanding things in this sort of space, it's not a complete understanding, but we can kind of see where the structural motifs are coming up by analyzing this correlation information. Okay, so that's the end of part one. And I want to take the rest of my time to not so much talk about the structures, but possible ways in which the plants are building these structures and using models to understand those processes and the decisions that the plants are making in order to make the flowers. Okay, so the question then becomes, how do you make a porous cell network? This has actually been a question that's been asked before, all going back to the work by Darcy Thompson in the seminal book On Growth and Form, which was actually first published in 1917. And what Thompson said was, okay, if you want to make a porous cell network, start with a dense packing of cells. This can be in 2D in this triangular packing of disks, or it could be in 3D, which would be the FCC lattice of spheres. And you can basically say, make each cell, I'll highlight one in blue, just stick to its neighbors through some adhesive mechanism. And all you really need is that the boundary of this packing of cells is going to grow faster than the cells will grow themselves. So it's like relative to the boundary, the cells are going to be shrinking. And if you do that, Thompson includes this sketch in his book and argues that this, which actually is a sketch from, this is from the stem of a banana. This could be, this complex network of cells could be constructed from this starting point in that you have this lobed cell forming the sort of vertex plus parts of the edges of the network, and then these concave voids turn into convex triangular voids. To me, this says that the basic ingredients you need to make these networks of cells are cells that that can change shape, but also adhere. You need to start dense, and you need the cells to grow slower than the boundaries growing. And I just want to note that this general thinking can apply in both 2D or 3D. Doesn't really matter, but I'm going to be talking about simulations, and simulations are much easier to do in 2D. So for now, all I'm going to be talking about is how to make these cell networks in 2D, but the general principles I think will be applicable to 3 dimensions as well. Okay, so step 1, we have to simulate cells that can both be sticky and change shape. So this is more complicated than it sounds, but the basic solution is with discrete element simulations, where each element is a 2-dimensional polygon with n vertices. And in order to simulate interactions, you just need the vertices of one polygon to interact in some way with other vertices. Here I'm drawing them as circles, and you can imagine that each circular vertex has some excluded volume given by its radius, which will repel other vertices when they come too close. And if you want to do adhesion, you just need to make them grab onto each other or adhere to each other with some short-range attraction. So then the more complicated thing is, how do you simulate the change of shape? And for this, you just need to ascribe different energies to the different geometric qualities of the polygons. So for example, our polygonal cells will have some area. We can ascribe a energy that will penalize deviations in the area of the polygon from some preferred value. Similarly, our polygon has a perimeter that's made up of, it's the sum of these little segments that connect to vertices. So we can say that each of these segments is like a little spring that has some preferred length L naught. And particularly when we're dealing with plant cells have a cell wall. So there's probably also going to be some curvature penalty. So we can ascribe a preferred curvature to each vertex, and we say that there's some energetic penalty to deviating from that preferred curvature. Okay, so that's how we make deformable cells. The next part is, how do we make a initial condition from which to start? This part's a little easier. All we have to do is seed the boundary with randomly placed cells, and then we just compress the cells until they sort of fill in the box. And I'll also say that this is all going to be in periodic boundaries, which means that cells up here are the same as cells down here. And I'm coloring the different contacts that each cell has as they're compressed. The average cell contact number here is 6, because most of the cells have 6 contacts, and that's going to be a good place to start from which we form our network. Okay, so now we have all of our components together. Let's see how this works. So this is from a simulation, and here I'm just coloring the cells based on their size so you can see them differently. I'm going to have an initially dense starting point. They're all going to be adhering to each other, and I'm just going to grow the boundary faster than I'm growing the cells, which means that I'm going to shrink the cells. And again, the system's going to live in periodic boundary conditions, so up here is the same as down here. Okay, so if we try to make our network, we'll find that it sort of fails miserably. We don't have a network, we more have an island of adherent cells that basically separate from the boundaries due to the appearance of large voids at the very beginning start of the formation of the network. So what went wrong? Well, the cells didn't really deform enough. You see that these cells here that are losing their contacts, if they just stretched a little bit more, they might be able to save their network. And also the network failed at localized places. And if you can actually see, this is where all of the 4-sided voids are. Now, 4-sided voids are sort of inescapable if you start with a disordered dense packing like this. So this is going to be something that that we're going to have to solve in order to get networks that look more like the ones that we see in experiments. So in order to save our simulations, we're just going to have the cells change their preferred shape over time. They'll reinforce their shapes by changing their preferred curvature in order to support more network-like forming cell shapes. And the adhesion is going to be contact dependent, which just means that if I lose a contact, my other contacts that I still have grow stronger. So the cell shapes are going to age with time, and the adhesion is contact dependent. And we're at 5:30, so I will just show quickly what you get when you add these ingredients together. And I'm just going to color the cells by their preferred shape parameter, which is a non-dimensional parameter that is the preferred perimeter, which is the sum of all the preferred lengths divided by 4 pi times the preferred area. This is just a nice way to quantify how much a cell has deviated from a circle, because if you have something that prefers, all the preferred geometric things are that of a circle, this number is 1. And I'm just going to color redder cells will be ones that are growing their preferred shape parameter. And so if we add in cell shape aging and contact-dependent adhesion, we see that we make a much more homogeneous network. And so we can look at what happens if we can vary the different parameters, like if we age the perimeter more or less, or if we age the curvature more or less, we can get different types of morphologies of networks. So up here, I'm not going to age the perimeter at all. So the fact that there are different colors just means that the shape parameters don't change during the simulation. And if I do this, you don't really get networks. You get networks on very large length scales, but most of the cells aren't participating in the network. They mostly just fall. They break all of their contacts because they weren't able to change their shape enough to support the formation of the network. If the perimeter ages a little bit more, you can start to see, okay, we get some networks, but it's still the network, like the length scale of the network is much larger than a cell. So you get these big bands of connectivity, and then you get these islands of connected cells that aren't really participating. Increasing the aging a little bit more, and we seem to get into now what looks more like the isotropic networks. And you say, oh, then we should be able to, if we age the perimeter even more, we should be able to get better networks. Well, that's not necessarily the case. If you go too far, you actually get something that looks more like a rubber sheet with a big hole poked in it. Like here, for example, the cells are all changing their shape, but they change it in such a way that it actually becomes more susceptible to these big rips in the center. So it seems like there's some Goldilocks parameters here that gives us the shape that we're looking for. So we're going to just compare what that looks like to a real sample from a real experiment. This is a tiny subtlety, and this is a long story attached to it, but I'll be comparing not to the spongy mesophyll of a flower, but that of a leaf, which looks similar, except that in leaves, the spongy mesophyll is layered in 2 dimensions. So you can compare to 2D things more easily when you look at the mesophyll in leaves and flowers, but the general principles are pretty much the same. And I'm also going to be comparing, so I'll take that image that I was just showing from a 3D micro CT scan of a leaf, squash it in 1 dimension, so I have a 2-dimensional image, and then I'll just do the same sort of correlation measurements that I was looking at from before with the 3-dimensional images, but now in 2D, and I'll just be comparing one network that I think looks pretty good, and then to a couple of test cases that I think we can all agree do not look similar to this structure at all, and we can compare what the structural features give us. And when we look at the correlations in Fourier space in 2 dimensions, we see that the structural features from the actual experimental mesophyll sample is this interesting sort of ellipsoidal shape with excluded zone at low K and at high K. And although the simulation images, nothing quite perfectly matches this, these 3 images are totally off, but I think here we have it. We have the beginnings of a little ellipse, so maybe it's just rotated along an axis. But I do think that this is evidence that we're getting some of the larger wavelength structural features in these selected simulations that totally distinguish this sample as more realistic than these other samples. But okay, so to conclude, we've looked at some very complex tissues that occur in the mesophyll of flower petals. We've seen that there's a large variety of structural features from different amounts of porosity, anisotropy, different length scales where different things are at play. But we're able to sort of wrangle the complexity of the different shapes by looking at their correlations and understanding what are the length scales in which the interesting physics is, the interesting structural features are occurring. And then if we want to think about how to build these tissues, albeit in simpler cases in 2D, we need a careful balance of adhesion, but also changes to the particle shapes. But it has to be sort of tuned to promote this network formation, and that will bring to bear what the actual flower petals have to, the problems that real plants have to solve in order to make these complex networks. So anyway, that's all I have to tell you today. Thank you so much for your attention, and I'd be happy to answer any questions.
Great. Thank you very much. If you have a question, go ahead and unmute and ask if you are a student. Students first.
Hey, hi, great talk. I have a question. So when you found that there are these mesophylls with similar motifs, does it mean that they have similar properties as a flower? How do we understand that? Because
yeah, right. So I don't have any like sort of metadata on the. Are you talking about sort of like different like things like
exactly this one? Yeah.
Yeah, so it's a great point. I don't have a lot of metadata on like, say, the typical surface area of the flower petal or something like that. So that's a great question. How these things percolate up to larger length scales is something I don't really know. One thing that we've talked a lot about with our botanist collaborators is that these samples that live, they're more porous and they're typically more isotropic. Those are, I think of them as like they're newer species, but they're later divergent from plants that didn't have flowers. So, and there's other research to suggest that the flowers, the flowering plants that have diverged more recently typically have smaller cells. And so there's potentially this convergence towards smaller cells, more isotropic structures, and more porous structures because those are cheaper, and you can potentially get a nice landing pad for a bug for half the carbon or something. So that's probably why they're living in that pocket, is because there's this evolutionary trend. But yeah, these samples, they're not related. They're not, none of them are really on the same tree or whatever branch of evolutionary space, other than the fact that they're all kind of newer. Other than that, I can't say too much more about the sort of related physiology.
Thank you.
All right, maybe one more question.
So it was a very nice talk. So let me ask you a very simple question. Since you're doing this airplane model for petals that say you're trying to use the least amount of material with the maximum amount of resistance, can you learn something from there? Why that's the choice? Or is just an observation, or can we figure out that was a smart strategy the way it's being done?
Yeah, that's a great question. This is what keeps me up at night. I think I can't say too much more than just that this is an observation, but I can say that the more porous samples are more isotropic, and they typically come from species that have diverged a little later on the evolutionary timeline, like I was just saying to the previous question. So maybe what we can learn is that this is somehow more optimal in the sense that it's being selected over a longer time scale. Like, after you have more iterations of the design lab of evolution, you converge to this better structure because it's cheaper. And I think it's also saying that the material that the flower petals are made of can support. I think it's interesting that they can support these porous isotropic networks. So these things can be made, and I think it's interesting to look at. So these are better in the sense that, or they're interesting in the sense that these cells are probably not so distinct from a material perspective than this cell, but this plant has learned that you can make them much more porous, so they're cheaper and they require less water to maintain. So it's I can't say anything more than that, but it seems like these. I think one interesting thing to look at would be mechanics. So we could just take these petals and like squish them and see how the mechanics differ. But I think if we assume that they're all basically mechanically the same in the sense that they all are petals that aren't drooping, the only difference is that these are more porous. So maybe this plant was just lucky enough to make a more porous structure.
Thank you.
All right. Thank you once again to Jack Treado. Now we will move along to Dr. Sweeney. if you are ready.
Great. Hi, everyone. For those of you who perhaps met me before, I was in the physics department at UPenn until pretty recently. I moved to Yale a year and a half ago, so I'm really pleased to be getting incorporated into the Yale community and this community. And thanks for inviting me to share my work with you guys this evening. So in trying to pick or decide on a topic for today, when I heard that Jack was going to be talking about his mesophyll stuff, I thought I would talk about the set of work that's most analogous in my group, and that is perhaps paradoxically this giant clam system. And so I've given it the perhaps provocative title that giant clams are perhaps the most efficient photosynthetic system on Earth. And, you know, I don't know that to be true yet, but I think we could make a pretty good case that they're doing quite well. And I'm going to outline that case to you this evening. And the first thing to notice is that this is a giant clam. The clam is about the size of a beach ball. And it's 10 to 15 feet away from the camera. I took this photo on snorkel. And it's sitting nestled among some reef building corals there. And the important thing about this clam and about this talk is that you probably know that corals are photosymbiotic. So that sort of biscuit color that they have is due to symbiotic photosynthesizing algae that they have in their tissues. But this giant clam has exactly the same species of alga nestled inside it, also doing a photosymbiosis. And so these two organisms are pretty visually distinct here. And I particularly want you to notice where we can see the algae in the clam, which is these sort of black stripes. The clam is indeed very black, very dark, where the coral is actually quite pale. And then, you know, there are other obvious differences here, which is like the clam is pretty flat and otherwise near spherical, doesn't look particularly much like a plant, whereas the coral is more branching and tree-like and more like what we expect our photosynthesizing organisms to look like. So the clam is both darker and flatter than the coral and seemingly sort of breaking a lot of rules about how to sort of pretend that you're a plant, which is unusual. And then, so in addition to that, where the clam surface is not really dark, you see this sort of skin or literal skin. It is in fact the clam skin, this skin of sort of almost cloud-like patterned cells over the top, which the coral also does not have. And so I started thinking about this system when I was a postdoc, and originally I just sort of wanted to crack the puzzle of what is a giant clam doing with an iridocyte. So those colored cells that you see over the surface are in fact skin cells that impart structural color, which you're seeing there. Okay, so that's sort of my entree into the clam system. And I want to now take a sort of a sideline into photosynthesis and just sort of note that photosynthesis gets a really bad rap for being an inefficient way to do solar energy. So if you don't work in this area, but are sort of like vaguely aware of sort of options for solar energy, you probably have this received notion that photosynthesis is very inefficient and is never going to be sort of a going concern for solar energy. And the reason we think that we understand this is because in high light terrestrial systems, and the reason photosynthesis gets inefficient in general is because there's this sort of fundamental mismatch between rate processes. So photosynthesis starts with the absorption of the photon and an excitement of a central pair electron on the magnesium of a chlorophyll molecule, and then a bunch of other stuff happens, but ultimately you have to match the rate of your electron excitations to the rate of thermal protein diffusion in order to turn the energy of this absorbed photon into biological chemical energy. And of course, the quantum process can go absurdly fast, and diffusive thermal process is stuck being several orders of magnitude slower. So if your solar flux is such that you can do these electronic excitement events really quickly, there's just no way that you're going to get protein diffusion to catch up. And it's really this rate mismatch between thermal and quantum processes that you can't fix. Okay, so in very low light, this isn't a problem because photons come in at approximately the same rate that proteins can sort of diffuse the absorbed energy away, and there's no problem. And this slide always goes a little, okay. So then universally what biology does in high light levels is just shrinks the number of absorbers. If you make fewer absorbers, your sort of apparent flux rate is much lower and your proteins can keep up and there's no problem. And of course, if your proteins can't keep up and you have all these excitons generated that can't be pulled into sort of the chemical energy processes, you get all this photo damage and your chloroplasts explode and you die. So there's going to be a lot of pressure to not have more photon absorbances than your protein diffusion can deal with. And so it's very easy for plants, and they're very good at shrinking their absorber when they even sort of smell too much energy starting to come into the system. And so here's what that looks like. So this is the common sort of algal culture Spirogyra, and they have these very beautiful helical chloroplasts along these filaments of cells. And in low light, those chloroplasts those helices get this really high pitch and they get really fat and they catch all the available light. And in high light, the pitch stretches out and they shrink. But importantly, now the power input to the cells on the left and the cells on the right is the same. So both cells are having their power requirements met. One is doing that by catching all of the ambient light in an inflated chloroplast, and one is doing that by catching much less of the ambient light in a shrunken chloroplast. So if you're a single cell, hooray, you solved the problem, move on and have a nice day. And we see this reflected now when we actually do an accounting of where the energy goes when we think about the solar resource. So the important thing to say, so I should also explicitly say, once that exciton is generated in photosynthesis, biology is exquisitely efficient about getting that energy off the chlorophyll and into the plant's chemical energy store. So chloroplasts can be greater than 90 or 95% efficient about taking excited electrons and sort of moving them through the pathway. All of the inefficiencies or apparent inefficiencies in photosynthesis come prior to that in just either not absorbing the light or in dissipating the energy of absorbed light into some other pathway to avoid photo damage. And so if we go ahead and look at the accounting of this, this initial loss of more than 50% of sort of incident solar energy comes from either the band gap problem. So obviously chloroplasts have band gaps like solar cells. So we're either going to not absorb it in the first place because our albedo is too low, or we're going to chuck it into the band gap. And then the other major source of loss is what's called non-photochemical quenching, or biology has lots of ways to dissipate the energy of an absorption event into other non-productive pathways, again, because it's so critically important to avoid photo damage. And then if you look at real systems, and then the rest of it, the other sources of loss are all quite small. And not real contributors to our sense that photosynthesis is inefficient. And if you look at real systems, you find the same thing, that basically you're losing sort of light relative to the solar resource from either the fact of having a band gap or the fact of not absorbing light in the first place. And then real systems lose a lot of the energy to these non-photochemical quenching processes. Okay, so if So how does it work then to go, so you have this single cell logic to just avoid absorbing the light, which works great for single cells, but then eventually, you know, biology and evolution cared to evolve multicellularity, evolve ecosystems where it presumably becomes important to start using more of the resource per surface area than cultures of single cells presumably do. So how does that work? And so now we have some physics problems. So if we want to absorb more of the light, it would seem obvious just to put sort of more cells in the path. But that's a big problem because now, you know, absorbance works as an exponential decay. And there's a pretty narrow band in which these cells are happy. So now by sort of just putting more cells in the path, the ones at the top are now, you know, over irradiated and shrinking their chloroplasts and doing all of that fluorescence loss. And then very quickly, you know, it takes only 2 or 3 or 4 cell thicknesses before there's not enough energy left in the beam to support life. And everybody sort of below 4 cell depths is in the dark. So, you know, biology, or, you know, we ultimately need something like a solar transformer to take the available energy that's coming in at too high of a flux to be useful, and basically use geometry to spread it out into fluxes that can be used efficiently by sort of the downstream absorbers. And then we have another problem, and this is sort of the relatively new stuff that I'm kind of excited about because we only recently realized that this is also a problem. So it turns out that the same number of pigments in a solution, so if I have a dilute solution of chlorophyll, that is not physically the same thing as taking the same molar mass of pigments and packaging them up into cells. And that's basically because if we think about sort of the beam through a cuvette and dividing that beam up into sort of little pencils of light, the absorbance of the system on the left here, where, so if I think of my little chlorophyll pigments as little cubes, and I divide it up so that I have the sort of same number of cubes per pencil of light in my beam, that is not the same thing as taking the same number of cubes but packaging them into cell analogs and then allowing those to have a distribution in space. So the absorbance of the left is not the same as the absorbance of the system on the right. And you get something like this, where the difference between the two, or the ratio of the two absorbances, goes as 1 minus, this is the transmittance of a particle or a single Q, over the log of 1 over the transmittance of the particle. And if we think about absorbance, which is a little bit simpler, this is the relationship that you get. So this is this graph is plotting the relationship on the left here, but in terms of absorbance rather than transmittance. And we see that the more that one of our sort of single pigment particles absorbs, the worse my suspension does relative to my solution. And so here is absorbance spectra of, in this case, a suspension of single-celled algae, and then those same single-celled algae blown up into a solution. And we can see this flattening that results from this relationship here. So this is algae, and this is the same thing for red blood cells. It works. And so we have this other problem as we get complicated, or as we generate more and more packaged and complex absorbing structures in biology, the more a given photosynthetic pigment absorbs, the worse it performs if my goal is to absorb all the light out of the system, which is yet another source of inefficiency. So I have some fundamental, pretty basic physics problems to solve if I am a single cell who would care to become multicellular and complex and solar powered all at the same time. Okay, so this brings us back to the Clam, and the way that I would argue the Clam has actually solved both these problems, the exponential decay in a bucket problem and the absorbance flattening problem that multicellular photosynthesizers have to figure out. Okay, so zooming in more carefully to the clam, these sort of zoomed in medium zoom photos start to show us that indeed the clam is so chock full of these algae that the tissue has become black, and then the green sparkly stuff here, or the blue sparkly stuff here, are these structurally colored iridocytes over the top. These iridocytes, here's a TEM of the iridocyte cells. The diameter is around 8 microns, and we can see they're roughly spherical with these protein platelets that have a thickness of about 80 nanometers. So the geometry is correct. So we basically have a Bragg stack, an optically or a visibly resonant Bragg stack templated inside a micro lens. So platelets smaller than the wavelength of light in a sphere, somewhat bigger than the wavelength of visible light. And these things lay over the top of these thick layers of absorbing algae. When we analyzed clam tissue, so if we take a plug of the giant clam tissue and we put it in a double integrating sphere experiment to measure where the light goes, we find this striking result that no matter what the different, so we see these different sort of visual patterns of iridocytes over the surface of the clam. You might think they all behave differently. When we put them in this double integrating sphere experiment, we found that they're strikingly identical to each other, and they have strikingly high and flat absorbance. So whatever else the heck is going on, it seems to be universally true that clams have extremely high absorbance and extremely flat absorbance, and very little of the light is lost. So the sort of prettiness of the clam patterns is a little deceptive, because ultimately you're seeing a very low degree of loss over a background that's extremely black. We also did an experiment where we surveyed clams in the field on coral reefs in Palau. And this is just sort of a synopsis of this photographic survey that we did. But one of the many analyses we did is we analyzed natural clams for albedo in the sense that an astronomer would use the term albedo. And so we characterized clam albedo in this photographic survey across many species and many reefs. We have something like 800 clams represented here. And what we found is indeed, not only do clams have universally low albedo in nature, so that's the red dots here, albedo actually goes up with depth. So I told you that cells are universally really good at avoiding light when avoiding absorbing light when it gets too bright. And so here, this zero depth is like, centimeters of water depth at 7 degrees latitude on coral reefs. So these clams are being blasted with light at flux near the solar constant, and yet they are only rejecting around 5% of it. And they actually absorb less as you go deeper and the light gets less intense, which was peculiar. And then if you compare to coral in the same photographic transect, the corals look more like trees. So this albedo of 20 to 30% is what you would expect for temperate forest, for example. So that was pretty striking. Okay, so what's really going on in the structure of the clam is that if you take the iridocytes off the surface and look inside, the clam has organized, I guess I don't get a video. The clam has organized, there we go, its algae into pillars. So the algae live in these vertical micropillars with the iridocytes over the top. Okay, and my videos are choking here. There we go. Okay. Okay, so the algae live in these, have been templated by the clam into these vertical micropillars. There are iridocytes, which are the blue coordinates here over the top. Light comes in to the surface, and then we ultimately understood the optical behavior of these iridocytes in collaboration with astrophysicist Sanazva Hedinia, and we utilized her discrete dipole approximation model of Saturn's F-ring particles to try to understand how these special iridocytes scatter. And what Sanaz's model showed us is that when light comes into the Clam's iridocytes, it scatters. It's strong forward scatterer, but the light does not go sort of forward toward 0 degrees as it would through a microlens. The presence of the nanostructure in the sphere actually causes a divergence of the beam to 10 or 15 degrees away from forward. And this happens in a very strongly wavelength dependent fashion with sort of the juicy blue and red wavelengths where the chlorophylls absorb being more strongly scattered into this forward cone than the sort of intermediate wavelengths. So that was very striking. And what we realized was that if we think about the geometry of these pillars, which is that they have openings between the pillars, so the gaps between the pillars are about 150 microns, and they extend to several hundred microns or millimeters into the tissue. The way that you would sort of have a transformer function, that is to say, take the flux present on the horizontal opening of the opening, the horizontal surface of the opening, and spread it out over the vertical surface of the pillar, you would need a phase function or a scattering probability with angle that looked like this. And what Sanaz's model showed us was this with wavelengths. So basically what the iridocytes are doing is becoming something like a transformer and taking the light impinging on the horizontal opening between the pillars and doing the correct angular transformation to get it evenly coating the vertical surface of the pillars. Okay, so that solves this exponential decay in a bucket problem that multicellular photosynthesizers will have. What about the flattening problem? Okay, so Doysens, who first discovered this flattening issue in the 50s, tells us that we can convert between the sort of solution pigments and packaged pigments through a factor F, which is basically the ratio between the length of your cuvette that you've illuminated in your solution and the sum of the lengths of your packages in solution. So that's Doysens' F parameter. It works fine in our hands. So F of 4.33 is what we expect for a solution of Clam pigments packaged into 10 micron Clam photosynthesizing cells. And that works great in our hands. So Doysens' packaging model works great for Clam pigments into Clam cells. And then if we want to go to from Clam cells into Clam, it doesn't work at all. So here the left panel is showing, so basically our Clam pillars in our Doysens model say that we basically get an F factor of 2. So the tops of the pillars take up about half the length of the measured beam in the system. And so if we consider an F of 2 for our Clam pigments, we get nowhere near the flattening that we need to explain the Clam. And in fact, we need an F of 30 to get a really nice fit between the Clam's constituent pigments and that beautiful flat albedo we find for the Clam. So we're missing a factor of 15 in area, in sort of red area here, to come up with the difference between our solution of pigments and our Clam flat absorbance. Lo and behold, it turns out that it's about exactly a factor of 15 to consider the sort of long surface of the surfaces of the pillars in addition to the tips of the pillars. So that breaks Doysens' model. But what happens when you position an iridocyte here is that these vertical surfaces become effectively horizontal area available for incident light. So by putting an iridocyte here, you're basically doing the same thing as uncrumpling all of the surface area in these rugose villi or pillars within the clam, and sort of solving the problem that the better a pigment absorbs, the worse it does as you package it up into cells. And so this is another way of looking at this. This comes from a Monte Carlo ray tracing model we built of the clam, where basically if we say there's roughly a factor of 10 more surface area in the pillars than in the sort of apparent bit of clam area that the pillars fit under, you can look at sort of the histogram of the number of cells that absorb at a given rate, and you get this really lovely sort of transformation of the flux, where if you put all the clams in our sort of exponentially decaying layer, we have a lot over irradiated at the surface and a bunch more in the dark. If you simply reshape them into pillars, which is our Doyson's model, where light from the sun or in our cuvette is only allowed to hit the pillar tips, you have this sort of boom and bust cycle where the cells in the tips are seeing sort of the incident flux and everybody else is in the dark. And then when you add the iridocytes into the model, lo and behold, we get a lovely peak in our histogram right at a factor of 10-fold, which is how much more area we put in our pillars than under sort of the apparent surface. If we look at this as like a rate problem, this 10-fold sort of transformation factor does a really lovely job of moving all of the cells into sort of this over irradiated zone into a flux at which they can be happy and at that very highly efficient spot in their sort of energy conversion function. Yeah, and so anyway, so this pillared, so anyway, the pillar structure simultaneously solves the rate problem and the absorbance flattening decay problem that both cause photosynthetic systems to be inefficient in bulk. And then the only photosynthetic systems on Earth that have anywhere approaching the high absorbance and flat absorbance of the giant clam system are spruce or sort of boreal spruce forests. And lo and behold, when you sort of take a drone photo of a spruce forest and compare it to a backlit giant clam, the pillars of the clam poking up and the spruce trees poking up at you look very similar. And we started to speculate if maybe spruce forests, if clouds don't serve the same sort of radiance redistribution function for forests as iridocytes do for clams, and one is just a geometrically scaled down version of the other. So that'll be the next time you guys invite me to give a talk, I'll give that talk. So anyway, with that, thank you so much for the invitation, and I'd be happy to answer questions.
All right, thank you so much. That was cool. Do we have questions from a student?
I would have a question. So I was wondering where these patterns, so these patterns of pillars are also these iridocytes, where this actually comes from, or how this emerges in the first place?
Sorry, what emerges?
Like the pattern structure, like the pillar pattern, for instance, or also the distribution of these iridocytes?
Yeah, so the iridocyte distribution, we've worked really hard on that and come to the conclusion that it's basically boring. So what seems to be happening is that the algae layer is so absorbing. So when we perceive a pattern, we're perceiving visual contrast, not absolute brightness. And what seems to be happening is that the algae are so absorbing that any amount of sort of reflectance in contrast to that appears to us as very contrasty, although the absolute difference in the light levels is very small. And so as best we can tell, the reason the iridocytes look patterned to us is because there's actually pretty much every clam has essentially a monolayer of iridocytes, but there are locally dense regions where instead of sort of one iridocyte on average in the path, you have 2 or 3 iridocytes on average in the path. And so that gives you a big amplification in visual contrast, even though it's very low absolute additional loss of light from the system. And so, you know, we've thought about like, are there some sort of like, you know, repulsive particle active matter model causing those like locally dense waves and blotches and things, you know, and that could be interesting. But what seems to be biologically salient is just sort of having a monolayer of iridocytes over the surface. And from, you know, an absolute irradiance point of view, that seems to be what's going on. And from a visual perception point of view, we see, you know, this local, you know, locally saturated blue high contrast, and we seem to think that that's really appealing. And then as far as where the pillars come from, they're actually extensions of the clam's gut. So when we're looking at those pillars, we're actually looking at regions of the gut that have grown up through that surface mantle tissue, and what patterns those and makes sure that they stay at the correct geometry. We really have no idea. They sort of look like inverse gut microvilli. So, you know, maybe, you know, whatever pattern generator gives you gut microvilli could be giving you these pillars, and the dimensions aren't totally wrong. They just sort of grow out of the gut instead of poking into the gut.
Thanks.
Those are beautiful structures. I'm just curious of how much the structure is there because the algae borrowed the structure from the clam and just get along. But the structure is there for other reasons, structural reasons, or the way the distribution of the gut has to be than actually light absorption. So how much this sort of structure optimized for light absorption, but or any other properties that the clam needs in order to survive?
Interestingly, so the algae live in the gut. So the algae live in the gut, and they seem to be on some kind of conveyor belt. So if you dissect a clam open, like the actual stomach in the center of the clam is like this golf ball size structure that is just a bag of densely packed algae. So they actually poop algae, but interestingly, they seem to be really flexible in how much they rely on the photosymbiosis versus how much they're just like eating the algae. So the algae can actually be expelled by the clam completely intact and viable. They don't eat them, they just passage them through, and they're almost like local symbiodinium density generators or something. You know, in other contexts, they actually digest and chew up the algae. And there are subtle differences in how well developed this pillar structure is. Like it can fall apart basically, and it seems to sort of fall apart arguably in contexts where the clams like have enough to eat just by filtering and eating like a normal God-fearing clam. So basically the clams must have the algae. There seems to be a fair bit of flexibility in the system in how much the algae just sort of happen to be there and how much the clam is like lovingly spacing them and making the system super transparent and well structured. So yeah, so basically they're within clams, there's sort of continuum of they have this sort of like clotted looking unstructured algae in the mantle versus like the pillars are super regular and the iridocytes are super regular, and it just sort of looks optical. So this is a roundabout way of answering your question. So the clam seems to be able to lose its photosynthetic capacity that seems to be associated with just sort of a degradation of the overall structure and transparency of the system as a whole. So they don't seem to be doing much with it if they're not sort of actively farming it, but I don't know.
All right. Well, we are over time. So thank you once again to Alison Sweeney and Jack Treado for your talks. And we will see y'all. I think the next seminar at the end of February will be a career-oriented discussion for the students to get to ask some questions and some ideas about what you can do with a physics degree if you don't want to be a professor. All right. So thanks again. And that's it.
Thanks.